morning everyone, I'm Stephanie Abuel and we are the Group 5.
Hello everyone, my name is Karen May Cababa and I will be discussing some further points for gene therapy and the basic process of a gene therapy.
Hi guys, I'm Joseph Francis Okalinawan from Group 1 and I believe in the sayings that if you cannot do great things, then do small things in a great way. Thank you.
Hi guys, I'm Marcalen Lacerna and I'm here on this video to report about the germline gene therapy.
Hi everyone and good morning to all of you. My name is Rolf Lawrence T. Dulalia from Group 5, the aspects of gene therapy.
Hi Miss, I am Samris Emmet Kuban. My motto in life is, if it doesn't challenge you, it won't change you.
Hello again everyone, I am Wanda Altademery and I am here to discuss the bioethics of gene therapy.
Good morning everyone, my name is Maddox Jovian Macapagong.
Good morning everyone, I'm Stephanie Abuel and we are the Group 5. We're gonna tackle about the aspect of gene therapy. So for today, I'm gonna talk about what is a gene. But before that, we have here learning objectives. At the end of this lesson, the student should be able to describe gene therapy and its various forms, and assess the issue, potential benefits, and determinant to global health. What is gene therapy? So gene therapy is a potential method to either treat or cure genetic-related human illness, which focuses on the genetic modification of cells to produce therapeutic effect or the treatment of diseases by repairing or reconstructing defective genetic materials. How does gene therapy work? Gene therapy is designed to reproduce genetic materials into cell compensate for gene or to make a beneficial protein. Mutated gene causes unnecessary protein to be a faulty or missing. Gene therapy may be able to introduce a normal copy of the gene to restore the function of the protein. Researchers are studying a gene therapy for a number of diseases such as severe combined immune, hemophilia, Parkinson's disease, cancer, and HIV. We have here the history of gene therapy. In 1916, the concept of gene therapy was introduced, and in 1970, Friedman and Roden, author of a paper in Science titled Gene Therapy for a Human Genetic Disease. Also, in 1990, the first approved gene therapy in the US took place on September 14, 1990 at the National Institute of Health under the direction of William French Anderson. Procedure on a 4-year-old Ashanti De Silva received treatment for a genetic defect that left her with ADA, SCID, a severe immune system deficiency. So we have here some questions. So the first question is, would you subject yourself for a gene therapy without its 100% assurance of effectiveness or future negative side effect? My answer is no, because we don't know the consequence or how our body reacts into such a certain therapy. So the second question is, should gene therapy be limited to medical concern only could be used for aesthetic purpose? So my answer is no, it can be possible. For example, cosmetology, they can also help in gene repair.
Hello everyone, my name is Karen May Cababa and I will be discussing some further points for gene therapy and the basic process of a gene therapy. Further points for gene therapy. According to Fleisler in 2017, the Harvard Medical School and the Boston Children's Hospital research team were able to revive the basic hearing in a genetically deaf mice into a higher level of hearing into 25 decibels, which is equal to a whisper. When they inserted it into the cochlea, they used an improved gene therapy vector called the ANCAT, wherein it came from the Massachusetts Eye and Ear Ophthalmology, which it allows the transfer of the gene into the inaccessible outer hair cells. In 1971, the human gene therapy was made and inspired from the successful recombinant DNA experiments way back 20 years ago. It can be simply described as the insertion of a foreign DNA into the tissue of a patient. Now, to the basic process of a gene therapy. There are several approaches of a gene therapy that researchers are currently testing, including gene replacement, gene interference, and the gene addition. The first one is the gene replacement. It is the replacing of a mutated gene that causes a disease with a healthy copy of a gene takes place. The second one is the gene interference. It is the inactivating or the knocking out of a mutated gene that is functioning improperly. The third one is the gene addition. It is the process of introducing a new gene into the body to help fight a disease. So how does a gene therapy works? A gene must be inserted into another gene, wherein the another gene is a carrier called a vector, such as the viruses, since they can deliver the new gene by infecting the other cells. When viruses are inserted into humans, they must be modified so that they cannot cause a disease. Basically, a gene therapy is a promising treatment for a variety of diseases. However, it is still risky and it is still being researched to see if it is safe and reliable to use.
Hi guys, I'm Joseph Francis O. Kalinawan and I will be reporting somatic gene therapy. So in order for me to deepen the idea of somatic gene therapy, I will be answering 3 questions. What is somatic gene therapy? How it works? And why do we need this somatic gene therapy? So what is somatic gene therapy? So according to NIMSurgeon 1988, somatic gene therapy involves the manipulation of genes in cells that will be helpful to the patient but not inherited to the next generation. In addition, this therapy involves replacing the genes of body cells in order to prevent genetic diseases such as cystic fibrosis, retinitis pigmentosa, and other diseases. We can include cancers also. So these changes will not be passed on the subsequent generation because the gametes are not altered. So other one is, it is a way to prevent diseases without need for a cure. Scientists are currently doing research to find out on what diseases could be prevented by this therapy. So 2 diseases the scientists are currently working on are cystic fibrosis and retinitis pigmentosa. Unfortunately, this therapy has not been tested on humans. It has been tested on animals, but the result weren't too satisfying. However, research is still making progress. So if this gene therapy works, obviously many people will benefit. So how it works? For somatic gene therapy to work, the altered genes are transferred into the patients by means of adenoviral vectors. So what is adenoviral vectors? It is viruses that are manipulated so that they cannot reproduce. These vectors are transferred into the patient either ex vivo or in vivo. So what is ex vivo? So from its root word ex, so it is the indirect delivery of altered gene. In this process, the adenoviral vectors would be introduced into a delivery cell that was removed from the patient that would then multiply. the cells would then be inserted into the patient. The advantages of ex vivo is it can control which cells are altered and high transduction efficiency, while the disadvantage of this ex vivo is it has difficulty removing enough cells and re-engroupment them into the patient. So what is in vivo? From its word in, so it is direct delivery of mutated genes. So the adenoviral vector is directly inserted to the patient. What is the advantage of in vivo? So it can access virtually any site in patient. While what is the disadvantages of in vivo? So first is difficult control dosage. Second is difficult delivering enough therapeutic vector to target without toxicity to other tissues. Third one is the risk of dissemination in vector throughout body and contamination of germline. Somatic gene therapy can also be done either prenatally or postnatally. For early onset disease, it would make more sense to perform prenatal gene therapy, or it more advisable to have prenatal gene therapy so that the disease does not have a chance to develop or mutate. So, do we need somatic gene therapy? And why do we need a somatic gene therapy? Genetic diseases can be detrimental and burdensome to the person affected and can lower his or her quality of life. Many of these diseases do not have cures, especially cancers. So, therefore, it cannot be treated any other way besides gene therapy, and one of these is the somatic gene therapy. So, some scientists and medical professionals are opposed to somatic gene therapy because of the possible risk. Prenatal gene therapy can put the fetus and mother at risk. A possible risk is preterm birth. However, if somatic gene therapy can actually work, then the potential beneficial outcome of the therapy should outweigh the risk. Scientists are trying to find ways to make somatic gene therapy as safe and effective as possible. So before ending my report, I will ask my fellow civil engineer, would you subject yourself to somatic gene therapy or not?
Hi guys, I'm Mark-Allen Lacerna and I'm here on this video to report about the germline gene therapy. So, what is germline gene therapy actually? So, germline gene therapy is all about genetic modification of germ cells. So, germ cells. What is germ cells? So, germ cells is actually what we call the egg cell and the sperm cells. These germ cells that will pass the change into the next generation. So, from the word modification. So, modification is actually to renew, to renovate, or to change, or something like to make it better. That is modification. This kind of therapy actually allows for the correction of diseases causing gene variants to the certain passing down to the next generation. If you undergo this therapy, then your reproductive cells will be targeted. So the DNA will insert into your reproductive cells and it can cure diseases, but actually it has a side effect because this DNA will cause mutations. So your disease will be treated, but there will be another DNA mutation that will occur into your reproductive cells. So actually there is a side effect. And there are something like the scientist was actually tested mice. I mean the fertilized mouse eggs. So there are fragments of DNA that inserted into that fertilized mouse eggs, and actually that mice grown up and the offspring was new, and it was very good test. And I have another example here, a picture shown in the screen about the germline gene therapy. So here, as you can see in this screen, that's right guys. And once again, I'm Marit Alan de la Serna, and I hope we will see another on the next video. Thank you guys. God bless.
Hi everyone and good morning to all of you. My name is Rob Lawrence T. Dulalia from Group 5, the aspects of gene therapy. I'm going to discuss about stem cell gene therapy. So we all know the definition of gene therapy. Gene therapy is potential method to either treat or cure genetic-related human illness. So the question is, what is stem cell in gene therapy? And what is the role of stem cell in gene therapy? So, stem cells. Stem cells are mother cells that have the potential to become any type of cells in the body or in our body. One of the main characteristics of stem cells is their ability to self-renew or multiply while maintaining the potential to develop into other types of cells. Speaking of these characteristics, I have a short clip about the ability of stem cell. Stem cells can become cells of the blood, heart, bones, skin, muscles, brain, among others. There are different sources of stem cells, but all types of stem cells have the same capacity to develop into multiple types of cells. Stem cells are derived from different sources, two of which are embryonic stem cells and somatic stem cells, which to be discussed by Ed Cuban.
Hi, I am Samaris Emit Kuban. So I will be discussing embryonic stem cells and somatic stem cells. So embryonic stem cells are derived from a 4 or 5 day old human embryo that is in the blastocyst phase of development. So embryonic stem cells are pluripotent, meaning they are able to grow into all derivatives of the three primary germ layers: ectoderm, endoderm, and mesoderm. So in other words, they can develop into each of the more than 200 cell types of the adult body as long as they are specified to do so. Next is somatic stem cells. So these cells that exist throughout the body after embryonic development and are found inside the different type of tissues. These stem cells can divide or self-renew indefinitely enabling them to generate a range of cell types from the originating organ or even regenerate the entire original organ. So, somatic stem cells are present in many fetal and postnatal tissues and are also capable of self-renewal, and with appropriate signals differentiate into various cell types from the organ from which they are derived. That's all. Thank you.
Hello again, everyone. I am Wanda Altademery, and I am here to discuss the bioethics of gene therapy. So let me start with this question. What is bioethics? Bioethics comes from the Greek word bios, which means life, and ethos, which originally means custom. On the one hand, ethics is a branch of philosophy that studies the rightness and wrongness of human actions. On the other hand, bioethics is the application of the principles of ethics to the field of medicine and healthcare. Now, to broadly construed, bioethics as applied ethics is defined as the ethics of life. In particular, bioethics is concerned with the ethical issues that arise from the emergence and development in life sciences, such as biotechnology and medicine. Some of these ethical issues includes euthanasia, abortion, suicide, human cloning, allocation of healthcare resources, genetic engineering, artificial insemination, contraception and organ donation, and transplantation. So, apparently, there are ethical issues involved in gene therapy. And here are some of the inquiries. First, how can good and bad uses of gene therapy be distinguished? Second, who decides which traits are normal and which constitute a disability or disorder? And third one, will the high cost of gene therapy make it available only to the wealthy? And so, as we can notice, bioethicists address the morality of these ethical issues using appropriate ethical theories, such as utilitarian ethics, Kantian ethics, Christian ethics, and pragmatic ethics. For example, a bioethicist may argue that abortion is immoral because, using Kantian ethics, it treats human person as a mean rather than as an end. As in applied ethics, bioethics applies the principles of ethics to the field of medicine and healthcare. Now, some of these principles are stewardship, totality, solidarity, respect for persons, non-maleficence, beneficence, justice, and autonomy. As a healthcare professional, one must care for human persons in scientific way. But for one to be able to practically do this, one must understand the meaning of personhood. In essence, every human person has an inner worth and inherent dignity. Every person possesses these characteristics not because of what she has or does, but because of what she is, a human person. And as a human person, he or she must therefore be respected regardless of the nature of his or her health problem, social status, competence, a past action, and the like. Now, for these reasons, decision about health must aim at the maximum integrated satisfaction of one's needs biologically, psychologically, socially, and spiritually. And that's all for my part under the bioethics of gene therapy. We all know that science brings society to the next level. but always keep in mind that ethics keep us there. Again, I am Monica Autodemery. Thank you for listening.
So to continue about the bioethics of gene therapy, I am Maddox Jovian Makapagong. Could the widespread use of gene therapy make society less accepting of people who are different? The answer is yes. On a large scale of germline, gene therapy could result in selection of characteristics to improve the genetics of a population. The widespread use in germline gene therapy may make society less accepting of people who are different or who have a particular disability. Should people be allowed to use gene therapy to enhance basic human traits such as height, intelligence, or athletic ability? The current gene therapy research has focused on the treating individuals by targeting the therapy to body cells such as bone marrow or blood cells. This type of gene therapy cannot be passed to a person's children. So it should not be allowed to use gene therapy to enhance human traits because gene therapy could be targeted to egg and sperm cells. However, which could allow the inserted gene to be passed to future generation. This approach is known as germline gene therapy. Would you subject yourself for gene therapy without its 100% assurance of effectiveness or future negative side effects? For me, no, because in this case, gene therapy is not 100% safe and we cannot assure the effectiveness or future negative side effects. Should gene therapy be limited to medical concerns only, or it could be used for aesthetic purposes? I think it should be used for anything if it has a purpose, because gene therapy can be used for aesthetic along with the medical purposes. The application can be like in the form of plastic surgery.